Hi everyone, here we are at our final week for Biology 120 lab. You've worked really hard this term. You've done a lot of work. I'll still be grading over the next week in a few days. Um, and you have one more assignment to get turned in. So I'm going to talk about that in just a second. Let me do a quick review for what we did last week. We learned about the carrying capacity of ecosystems, and that means how many members of a particular species an ecosystem can sustain without damage to the ecosystem, without the extinction of that species, and that sort of thing. So carrying capacity is very important, especially for the Department of Natural Resources wherever you live, because they determine hunting limits and species that are going extinct and that sort of thing. You also studied cohorts, the different cohorts from the gravestone cards that you had, maybe males and females born in a certain time period, to see what happened to those populations. And from studying cohorts, you learn about what is happening in that population and what things might need to change in order to sustain a population. This week, you're going to be measuring population size. So how do you actually count populations? And they're counted in a number of different ways. We are going to look at 3 different ways. So transects are straight lines that you run through an area and count how many species as you go along that line. We are simulating this with a board that has different colored squares on it, and you're drawing lines, and then you're counting red, green, and blue squares along those lines to represent different species. Quadrats are just rectangles of area that you count in. So you mark off an area, and then you count how many of a particular species or a number of different species that are in that location. And you decide how big that quadrat is and which different species you will count. We will also be using the same board for the quadrats, and you're just using pipe cleaners to mark out your rectangle. And then mark and recapture, and this is particularly important for animals that move around a lot. Not so important for plants because, of course, plants stay still. But mark and recapture is where you mark or band certain individuals over a period of time, and then when you go back and recapture members of that species, you figure out how many marked ones there are and how many unmarked ones there are, and in that way you can extrapolate and figure out how many members of a species there are out there. I know in my area there's a man named Bill Hilton who lives at Hilton Pond. That's just his house and his plot of land, and he has been banding hummingbirds for decades now. He has all kinds of data on hummingbirds that he has banded and how many he has recaptured. He has all sorts of data on them, so that's really interesting. But that's done all the time to study populations, especially of animals that move. This week, a very good resource for you is the OpenStax Biology textbook, which of course is free. All you have to do is look it up, Google it, OpenStax Biology 2nd edition textbook. It's right there. If you look at chapter 45, section 1, this is the ecology chapter, it tells you all about why you would use transects for population studies or quadrats or mark and recapture. So please look at that section because it gives you a great bit of information about why you use these particular techniques, and you're going to have to answer those questions in the lab report. One hint for this week, it tells you to use a dry erase marker for your sheet that you have, the laminated sheet. I just used my wax pencil that I got with my kit, and it worked perfectly fine and didn't rub off when I brushed against it or anything, and it just wiped off with a paper towel. So just know that you can use that wax pencil for marking your transects and that sort of thing, counting, all that sort of thing. Like I said, you're going to have to figure out why you use the different population sampling techniques, the transects, the quadrats, or the mark and recapture techniques. and look at that chapter 45, section 1, and it gives you many, many, many good examples. It's all written out there for you. So please do that. So that is your one assignment this week. Week 8 is the least amount of work that you have to do, and that's because I have to catch up with you in all this grading, especially from week 7. All right, a couple of last things. You have to turn in unfinished work by Sunday, December 20th at 11:59 p.m. Please be aware of this. This is your last day to turn in any work whatsoever, any work. At that point, at 12 midnight, the course closes for you, and you cannot add any more work, and I cannot add any more work. So just be aware of that. No matter what the late policy is, no matter what, the course closes at midnight on Sunday, December 20th over to, Monday morning. Also, I would ask you to please do your course evaluation. That helps me out as an instructor, helps me learn from you, things that I do right, things that I maybe don't do right, things that you would rather me do, things that I could do to help you. And this is for future students, of course, but it's helping me be a better instructor. This also gives my team leader and my dean an idea of how I'm doing as an instructor. So please, please, please fill out your course evaluation for me and for this course. I would appreciate it so much. And I hope you have a wonderful and happy holiday season. Please be safe from COVID. I know everybody's saying that, but please do be safe during these crazy times. I'll keep you posted. I told you that I was in the vaccine trial, and I go tomorrow to have my antibodies checked. I will keep you posted. I'll send out an email message and let you know how that's going. And if I actually got the vaccine or I got the placebo, I'll let you know as I go along. I'll send out email messages to keep you updated. And with that being said, you keep me updated with you too. I love hearing back from my students. So please email me anytime. Let me know what you're up to, what you're doing. And I hope you have a wonderful week. Let me know if you have any trouble or you have anything going on. m.sigmon@snhu.edu. I'm here for you. I will answer your questions. I will help you with your issues. Have a great week.